Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. From Microbe TV, this is TWiP, This Week in Parasitism, episode 248, recorded on November 22nd, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about parasites. Joining me today from New York, Daniel Griffin.
Hello, everyone.
What you got on your tie? It's Friday, so it's got to be an STD, right? Gonorrhea.
I don't know exactly.
The gonococcus.
Is that like cocktail hour or something?
Yeah, the clap.
Also joining us from Fort Lee, New Jersey, Dixon Despommier.
And ditto, hello, everybody. Today was an auspicious day in the fact that it was cloudy and it was not warm. And yesterday was even more auspicious. It actually rained. So that was an event that hadn't occurred since late September.
Still raining now, supposedly. It is 4 degrees, yeah.
Well, it's not raining here right now, but it might pick up again.
It's cold, it's wet, it's raining where I am.
That sounds like your sailing season has come to an end.
Sounds like Glasgow
this
weather.
is winter sailing season. I'll let you introduce our last co-host, and then
also
I'll
joining us from
you some recent sailing.
Glasgow, Scotland, Christina Nowla.
Hello, everybody. I think I'm beating you all with the weather because it's been quite nice, freezing cold, but no rain.
I thought it's always cold and wet and rainy in Glasgow.
It's usually wet and windy. Cold, I don't know, I suppose that's relative. It's below freezing. Is that cold?
Yeah, it's cold.
It doesn't usually get that cold, so it's
anything
quite unusual.
Below 10 C is cold to me.
Okay.
Yeah, right now it's 4.
I've had to scrape the car today, so it's definitely cold.
Oh, that's cold. Yeah, no. So I was gonna tell Dixon that in the winter I race sailboats every Sunday. And I usually do that with my cousin. And there is this one guy, John Browning, who he's a professional sailor. He was a main trimmer for the America's Cup. He more Newport Bermuda races, Annapolis Bermuda races than anyone living. So when he's out sailing, it's you got to really pay attention. But I thought this last weekend I was going to go out. My cousin and I were going to sail. It was going to be relaxing. We were going to chat about politics. But there's some new kid, and he's one of these college sailers. And there's some stiff competition there. So we sail right through the winter.
Brr. I just say my hobby is fishing, as a lot of you know, and I used to fish all the way through the winter as well, no matter what. If there was ice on the water, but there was a little rivulet of water that you could see because it didn't quite freeze over, I would still fish. Now, if the sun isn't out, I don't go.
I used to do a lot of winter nymphing when I was out.
That's right. No, that's exactly right.
Yeah,
exactly right.
It could be quite productive. And also, the fish seem less skittish maybe in the winter. So I could go with my dog and she could be in and out of the river and doing her thing. And it's beautiful, beautiful.
Daniel, did you say nymphing?
Yeah. So, as Dixon well knows, 80-90% of a trout's diet is subsurface. It's not on the surface.
That is true.
And so if you're surface fly fishing, it's actually not as productive as if it's called nymphing. And so you actually put a little weight or use a sinking line, and you're actually going to get the earlier forms of the arthropods down there to where the trout, you'll often see it looks like they're rising, but they're not rising. It's the tail breaking the surface as they're going down.
That's right.
I've never heard that term. I've only been fishing once, so.
There's even a term called Euro-nymphing. It's a specialty, a way of handling
This
the
all
nymphs.
sounds too obscene to me.
Well, it depends on how you do it, but
these,
you're
like,
right.
Beautiful maiden, spirits of the water.
That's right. That would be naiading.
If you enjoy these conversations, we'd love to have your support. This is our fundraising season. You have two options here, actually. You can go to microbe.tv/contribute, or you can go over to parasiteswithoutborders.com. If you donate there, Daniel will match your donation. So just decide what you want to do, because we need more of this science communication these days. As you know, science is under siege. We have the nomination of RFK Jr. as head of Health and Human Services, someone who has zero qualifications and has contrary views to the health of Americans. And so, in fact, you should write your senators and oppose his nomination. I have a website to send you to, safecommunitiescoalition.org/RFK. You can put your little letter in there, and they have a draft already made for you. You put where you live, and it will send it to your senators. I have a solution for
this, by the way, Vincent. And
what's the solution? Dilution?
No, the solution is that only the Republican kids don't get vaccinated.
Well, red states had higher incident of COVID, right, Daniel?
Unfortunately, there was what they referred to as red COVID, because, early on you saw a lot of the deaths, hospitalization, morbidity in urban centers. But then, unfortunately, once vaccines were available, once we had a lot of therapeutics available, there was sort of this reverse phenomenon where a lot of the other areas were really hard hit. And it really was tough. I don't understand the messaging. There was a lot of uptake of the monoclonal antibodies, which, thumbs up. That was fantastic. 80, 90% reduction in progression, hospitalization. Unfortunately, no reduction in long COVID. Where in other areas where you had the vaccines, not only did you get that 90% reduction in hospitalizations and deaths, but we're also now seeing really significant impact, probably about the same, probably about a 75% reduction in the risk of long COVID, post-acute sequelae. So, and I think that, what is it? The solution to pollution is dilution. So maybe if we can dilute out the misinformation with accurate information from those of us who are not trying to sell you snake oil, then maybe we can make a difference. So, but we need your money so we can get our
Absolutely.
By the way, speaking of snake oil, Dr. Oz was nominated as head of Medicare, Medicaid services, right? What do you think about that, Daniel?
Unfortunately, as well, I think many of us, 3 of the 4 of us, right, Columbia faculty, there was actually a letter, which I think was signed by, maybe 10 or 12 Columbia faculty, basically calling out for the fact that he was knowingly misinforming people so that he could sell his products. He could sell his snake oil, right? So that I think that's probably what brings it up. So that's not okay to take someone, put him in the head of, Medicare, Medicaid, so CMS, who has a history of ethical laxity, of actually basically misinforming, lying for their own financial self-interest. That's not okay in my book.
Apparently, also owns a lot of Medicare companies as well, so that would benefit him.
Oh, interesting. So, yeah.
Well, I hearken back to that quote that I am often prone to say when this topic comes up, but it rings true virtually every time I say it. Mark Twain was quoted as saying that there's only one criminal class in America, and that's Congress. And everyone laughed, and then they stopped laughing when they realized it was true.
It's true.
Yeah. Yeah. I mean, I don't want to, make all our programs maybe it is okay. All our programs really are about combating anti-science and misinformation.
Yeah. Without funding, there is no science.
Yeah. Yeah.
And the funding all comes from government.
One of the things that RFK Jr. has said before is that he would like an 8-year pause on infectious disease research in the U.S. And, many of our programs are about infectious diseases, right?
Today
we're going to talk about a paper that involves infectious disease research. So
you bet.
To think that you could stop it for 8 years is the silliest of silly notions, okay, to put it mildly.
Is that even possible?
It reflects an ignorance of what infectious disease research is.
That's right.
Yeah. Well, this maybe that leads us right into our paper, which is where infectious disease research may actually be tapping into Alzheimer's disease,
right?
Which so shall we jump into it? Okay,
let's jump right into
it.
So this is why you're here. So today we're going to discuss what's real. I have to say it's a great paper, and so this is a paper entitled Toxoplasma gondii infection of Alzheimer's disease mice reduces brain amyloid density globally and regionally. And, I'm old school. I printed it out. So the authors are Catherine Yanes, Nathaniel Guanzon, Ricardo Azevedo, Damian Wheeler, Sunil Gandhi, and Melissa B. Loden. And a lot of these authors are from California, right? So Department of Molecular Biology, Biochemistry, University of California, Irvine, California,
right.
Institute for Immunology, University of California, Irvine, California, and also Translucence Biosystems, Irvine, California.
So it helps to clarify the title a little bit, and that is that we're not talking about globally and regionally geographically. We're talking about it in the regions of the brain. It's a little, for anyone not really up on this literature, perhaps they would be a little bit confused about the how did you get mice out of a regional? No, we're not going there.
yeah. Which part of the world does it help the mice? But no,
it just confounds the issues. No, but they do talk about the brain as a global issue because it's got so many parts.
Yeah, no, and I think that's going to be an area that we'll touch on. I think they could have just gone away with Toxoplasma gondii infection reduces brain amyloid density, and but there is some parts where it becomes interesting for those sort of deeper in the weeds about the different areas of the brain that they're going to look at. So
right.
So maybe we should start off just by talking about Alzheimer's and the current theories about what causes Alzheimer's, what's going on. So I'll start off with just the clinical perspective. Like what is Alzheimer's? So Alzheimer's disease is a form of dementia. And one of the things clinically we make a distinction from is memory loss. Sort of this memory loss that we expect, unfortunately, as people get older, just forgetting things, compared to dementia. Dementia is really a cognitive dysfunction. It's confusion, disorientation, trouble, activities of daily living. So Alzheimer's is a form of dementia. Okay. And it's one of the, we'll say, neurodegenerative forms of dementia. So breaking them in, we have vascular dementia, right? So someone might have major or minor infarcts, ischemia, damage to the brain, not getting enough blood supply to different parts of the brain. So we have our vascular dementias. And then we have a whole group of these neurodegenerative, where it seems that the brain itself is degenerating over time. When we get to Alzheimer's, there's been, I'm going to say, three major theories on types, pathogenesis, what's driving it. And the number one that people have probably heard most about is the amyloid theory. And it's interesting because they talk about camps. They talk about people who are in the amyloid camp. They talk about people who are in the tau camp. So the other is there are these tau that form. And then there's this inflammatory, the idea that, those are all just sort of markers of an ongoing inflammatory process. And if people had any comments sort of there before we go moving forward. It's interesting the way there's camps here. Like it's like it isn't a bipartisan. You got like the partisan here. You got one side of the aisle, the amyloid people, they want all the funding. You got the tau people, they want all the funding, and then you've got, I don't know, is this the Green Party, the inflammatory people?
Only if there's pus coming out, I guess. You also might want to add that Alzheimer's disease is on the rise, and I believe that's a global problem. It's not just a regional problem. It's wherever you go, it's increasing. So
I suppose that's got something to do with the aging population.
Mm-hmm. It might. It very well might. That's a good point, actually.
Yeah. Is it not, I presume you're meaning Alzheimer's in general and not early onset Alzheimer's that's on the rise?
Is that another camp?
I don't know. I'm a parasitologist.
Daniel, it is clear. I mean, Alzheimer's is something in general associated with advanced age, like as people get older. So as you have an advancing population, people stay alive long enough, then you can start to see dementia and some of it. And there's a couple things they go through here. So just, so the idea of the amyloid hypothesis, which we're going to talk quite a bit about today, is this notion that the amyloid plaque accumulation itself, like the amyloid itself, is toxic. And so the amyloid accumulates, it causes neural damage, it triggers the inflammation, it then leads to the cognitive deficits, right? So that's going to be a lot of this paper is talking about, so then, you would think like, well, if there's some way to decrease the amyloid, then we can decrease the neurotoxicity, we can decrease the inflammation, we can make folks better. And as mentioned, this is a huge problem affecting a lot of folks as they get older. And they start off with this background. And there isn't a lot of data in this paper, to be honest. There's two figures that we'll go through. That's right. But there's a lot of background context. So one of the things they point out is, and I'm just going to read this straight from the intro here, recent studies have shown a link between infection with the obligate intracellular protozoan parasite, Toxoplasma gondii, and protection from Alzheimer's dementia-associated pathology in mouse models. And then they mentioned that T. gondii is estimated to infect 13.3% of the U.S. population and up to 1/3 of the world's human population.
So it would seem a very straightforward study to say do the people who have toxo get AD?
Correct.
And there's certain areas of the world, right? Like in France and Brazil, where 80-90% of the population has infection.
Do they have AD in France?
Of course.
So, yeah, they do.
They do. They have over 1 million cases. I did some background reading on this just to see if there was a relationship, and it turns out, France's population is about 63 million. And they have 1.6 million cases of Alzheimer's. In the United States, it's 360 million, and we have 6 million cases of Alzheimer's.
So
and
in France,
it's on the rise in both places.
In France, it could be that those million cases are the people that don't have toxo.
It could be. It could be. I don't know if they ever checked or not, but
that's a pretty straightforward study, right?
I think they've looked at it. I don't think there's a correlation. I don't think there is.
Yeah, I think in the discussion they talk about that. Yeah.
I mean, the other, and we'll get into, is does it matter when you get infected?
Yeah, exactly right.
Yeah,
right. And there are other agents they mention as well that also cause a reduction in amyloid. So, Daniel, what is amyloid?
Yeah. So what is amyloid?
I got sidetracked here because amyloid builds up elsewhere too, doesn't it? It builds up in other parts of the body as well and causes different diseases. I got a little bit sidetracked.
I mean, is it a protein? Is it a fat? Is it a polysaccharide? What the heck is it?
So it's a protein. Yeah,
it's a protein.
It's a protein that actually builds up. And it can collect as Christina points out, not just in the brain. It can actually collect in a lot of other places as well.
Why is it called amyloid?
Oh, that's good. So what is the etymology of amyloid?
Yeah, I mean,
every time I hear, starch and cellulose-like. I think that's where that's from, isn't it?
Look at that. And so there's an enzyme called amylase, which digests starch and glycogen. And if people don't know what the chemical signature of this is, they're wondering, how does that build up in your brain and how does it interfere with transmission of neurons and that sort of thing? So I'm glad I asked the question rather than I was asked the question.
Me too.
No. So it does actually mean starch-like, but it's a protein, right? So where starch protein would be carbohydrate, so it's a starch-like protein.
Is this a repeated protein, like a polyprotein?
So it is interesting. Like, is there a connection here between like amyloid and misfolded proteins of like a Creutzfeldt-Jakob, which almost has like this PCR replication? We don't think so. It's not like what are those the replicator things from
prions?
So.
Or prions, depending on where you're from.
I mean, Stanley Prusiner thinks these are all, in the end, misfolding diseases, right? Because the amyloid is misfolding and the tau is misfolding to make these plaques and tangles. So they have a similar underlying mechanism.
Yeah, I mean, tau is also a protein as well. And so the same sort of theory there, that if you can get rid of these abnormal, toxic, misfolded proteins, you can have some benefit here. And so the interesting thing, we sort of get back to some of the things that we've talked about is, so does infection with T. gondii actually impact learning, memory, behavior? And so they point out that there's been some prior work where they've done T. gondii infection of this ED, so this Alzheimer dementia mouse model, so Alzheimer's disease mouse, and they've actually shown improvement in learning and memory deficits, and that this actually persists after infection. Follow the mice out for almost a year, right? Which for a mouse, that's like an eternity.
Exactly.
Right. What is it? My cousin says, like, if two people date in college, it's like you multiply it by 7. It's like dog years. So, mouse, they need to have, what are mouse years, right? It's going to be like
2 years on your toast,
right? I mean, mice live, 3 years? So
max, that's max.
Right. So that's like each mouse year is like 30 of our years. Right?
But interestingly enough, if the mice are kept on a minimal caloric diet, they live longer than their litter mates that are allowed to eat anytime they want. So being normal weight, not just overweight, but just normal weight, appears to be a pathological condition, which eventually ends up shortening your lifespan. That's a bizarre finding because there are a lot of people out there that believe that so much that they look like they're walking skeletons.
I mean, that's another one of those that might be true in mice, but we haven't necessarily seen that in people.
So another question arises.
I don't even know how you'd try it. How do you test it in people? You can't infect people with toxo, right?
Yes, you can. Oh, yes.
Well, yeah. So this is different. There's a group in Japan, right, which sort of almost like a cultish type group that believe this is how they're going to live forever, and they incredibly limited caloric intake.
Yeah.
And we'll see.
So, right. Dixon, you can infect people with toxo?
Under extraordinary conditions, you can. If they're in a state of ill health where no matter what you do in the known medicine world, they're going to die, then they often approve protocols for extraordinary experiments like this one.
So, Dixon, if you get toxo as an adult, what are the consequences?
Well, you get you don't get a learning deficit. There are no, as far as I know, there are no permanent effects unless it reactivates as an adult, and that came out during the initial AIDS outbreaks, and that diminished your immune response against everything, including Toxoplasma.
But if
a lot of AIDS patients died of encephalitis due to toxo.
But if you have a healthy person who does not have toxo, and then they get infected at age 50, will it become a chronic infection?
Oh yeah, well, not. It depends on how you define the infection, because it gets into this bradyzoite stage, and the bradyzoites are not dividing. So
should we go back and do this? Like, let's do basic toxo for our listeners.
We should probably do. We did that once already, but we can do that again.
Yeah, so let's start off. How do you get toxoplasmosis? Do you have to own a cat?
Well, there are two ways. One is that you eat the cat, and the other one is you take very good care of the cat. No, I think there are the primary way of the reason why the French have such a high rate of infection. The prevalence and incidence rates are very, very high, is that they have a set of cultural dishes and cuisines that if you don't eat them by the time you're 2 years old, you're not considered French. And one of them is steak tartare. And steak tartare is free of toxoplasma, by the way. Steak, as long as it's steak, it's okay, but they don't use steak. They use lamb, and they use mutton. And those animals are not free of toxo. And so a lot of toxo is transmitted by the ingestion of raw meat. In nature, this is, by the way, the most widely distributed protozoan infection in the world. The next most prominent is the one we had last time for a case history for Trichomonas vaginalis. And then I guess malaria is third, but this is probably the world's greatest successful protozoan parasite because, Vincent, if you acquire this as an adult and you have an intact immune system, you'll probably have 6 weeks of mild symptoms, a few swollen lymph nodes maybe, some fever. As the organism distributes itself throughout the body, thank you, macrophages, for picking me up and dropping me somewhere else. It's like the taxi cab for toxoplasma.
But we should say ingestion, right? It tends to be an oral ingestion.
Oral ingestion is the only way you're going to catch this. It's not a sexually transmitted disease at all. So you get the infection by ingesting the bradyzoite stage, which is a dormant stage in muscle tissue, usually. But of course, if you eat brain or if you eat other organs as well, you have an option for getting it that way as well. But it's mostly through the ingestion of muscle tissue. So then the organisms are activated by passage through the stomach. And as they enter the small intestine, they receive environmental cues to sort of wake up, because now you're in the right place to start the next phase of your infection. The bradyzoites change into tachyzoites. Tachyzoites are the stage that we're used to looking at, these sort of banana-shaped, crescent-shaped organisms. And they can penetrate cells, and they penetrate the cells in the small intestine, and they set up an infection in the lamina propria, and it's loaded with all kinds of phagocytic cells, and some of them, like neutrophils, I believe, can actually destroy toxoplasma tachyzoites, but macrophages don't. And in fact, John Boothroyd, a friend of mine who worked at Stanford on this, has investigated why that happens. And it's because of the way it enters the cell and the kind of membrane it induces around itself to prevent the fusion of lysosomes. And as a result, the organisms are not destroyed. And that's one of its key survival tactics, is to avoid death by phagocytosis so that you can be distributed throughout the body. The other remarkable feature about toxoplasma is that there are no limits to the kind of cell it can infect. Now, in This Week in Virology, of course, we talk about all of the receptors for various tissues or for various cell types, and how specific some viruses are, at least for those cell types. Toxoplasma is not, it has no bounds with regards to entering and leaving cells as well. In cell culture, you can watch this happen all over the place. The cells are still alive, so it can punch a hole in the cell membrane, and in going through, the hole that it just creates seals up, allows the tachyzoite to go inside. And if you leave it in culture long enough, of course, it will replicate. And, you can use any kind of cell that you want. You're free to study this in any cell culture. And fibroblasts are good to start with. Okay, so in your body, this is happening all over the place, and you can imagine what effects this might have, because as the organisms replicate inside of a cell, just like with malaria, and they're related to each other, they're both abicomplexa, out come the tachyzoites to look for another cell. And in doing that, it destroys the cells that they were in. And that could go on for some time. That could maybe, if your immune system is active and your IL-12 is active and you've got lots of ILs there that are not ill, but rather helping the cells around them survive, then eventually, with the help of interferon and several other immune inducible responses, including immunoglobulins, the tachyzoites are brought under control. And when that happens, in most cells, they die. But in the brain and in selected other tissues, in the eye, for instance, also, the choroid plexus, the organisms can live for a long time. They inhibit immune responses against them by a very simple technology. They just stop dividing. They said, I'm not going to let you see me because my metabolism will go to almost zero. And those organisms transformed from tachyzoites into bradyzoids, which is a rounded up form of the banana-shaped tachyzoids. But the moment you eat the bradyzoids in the form of a cyst, they reactivate and become tachyzoids again. So the other thing about this organism is that it's very specific for its hosts with regards to reproduction. It's got a male and female equivalent, and the only group of animals that it can reproduce in are the felidae, so cats of all kinds. In fact, all cats are susceptible to both the tachyzoid stage that it eats from an animal that it just killed to the sexual reproductive stages, which never leave the animal and go in and disperse throughout the tissues. What that stage does is it fuses, going from a haploid to a diploid stage protozoan, and then they produce two internal spores. And the oocyst, which it's called the oocyst, and that name was also used for the sexual stages of Plasmodium as well. That stage is then excreted into the environment. So another way to catch this infection is by accidentally eating some cat feces contaminated with the oocysts. And they almost instantly, upon hitting air, mature into the infectious form that's then viable for some time. Of course, if it dries out, it dies, and that's the end. But carnivorism and scavenging is the way this organism finds its way around the world. And I think that's enough for the biology. The biology is very complicated. Once it gets inside of a cell, it's very virus-like in terms of its ability to interfere with normal host processes and even interacts with host genomics in a way that is reminiscent of the way Trichinella interfaces with its host genomics to cause the host to produce proteins that it never would have produced otherwise, at least under those circumstances, and enables the parasite to live a long and non-reproductive life until the next host comes along. And all you have to do is die in order to transmit this to another animal. And that's the real crux of the whole situation. Most infections don't depend on death of host in order to transmit itself from one place to the other, but Toxoplasma is famous for its ability to survive as these bradyzoites in tissue until something comes along and eats it.
How many human beings are infected? We're really kind of a bystander to this sort of
that's right. We're a dead end. That's right. Except we're not a dead end necessarily.
I guess we get eaten by a lion, right?
Well, no. There are cultures on the Himalayan mountains, mostly Buddhist cultures, which actually dismember a person who has died and put them on these platforms for the vultures to come and feed on the carcass to recycle the body back into nature. And other animals will scavenge on those parts as well. And when they do, of course, they have the option of acquiring Toxoplasma as well. You can also catch it from the mother. So there's a congenital toxoplasmosis as well. Yeah. And so that's a third way of catching this. And it's the one that causes the most damage in people. An adult can usually fight off this thing. If you're an AIDS, suffering from AIDS, then the answer is you can't. But if you're an immunocompetent person, then you can. But a fetus, especially in its first and second trimesters, has got no immune system, basically. So the parasite can take advantage of it. And eventually the parasite ends up in brain tissue, as well as other tissues. And the one most affected, of course, is the development of the brain in the fetus. And some horrible cases of congenital toxoplasma are available online for you to become absolutely repulsed by the damage that a very small organism can cause to a very innocent bystander, basically, that had nothing to do with this at all. The mother ate a piece of raw meat or had a pet cat that, she was not taking as good a care of as she should have, probably.
So, Daniel, tell us what they did in this paper.
All right, so please. Let's go into the paper.
Let's return to reality here.
Yeah, no. This is reality, right?
Well, that's true. That's true.
Clinically in people, it's oral. Unless it's from mom.
Yep.
Usually presents a mono-like illness, but then you end up with these intracellular creatures in the brain, and in most cases, and so that's where we're going to go. So let's get back to our paper. We've gotten much of the. I think the other thing we should probably mention in the paper, we're still in the intro here, is that the impact of the T. gondii infection in mice is different, whether you're dealing with your lab BALB-C, right?
Yeah,
probably some listeners are familiar with the little white BALB-C mice versus the wild type or even the Black 57 mice. So actually, it depends which mice you're infecting, right, as far as the impacts that you're potentially going to have.
Also, the mouse model is weird, right?
It is, because there's actually this special Alzheimer's disease mouse, right, which again changes things because you're doing the infection in a particular inbred, genetically modified mutant mouse, which you can buy from the National Institute of Health Mutant Mouse Resource and Research Centers. I'm sure they're going to get shut down.
Then they're transgenic mice that overexpress a human altered amyloid protein. Okay, so these are mutations in the amyloid gene that are known to predispose to misfolding. So they put 5 copies of the mutant genes into the mice to try and, because mice don't normally develop Alzheimer's.
Yeah, they have these 5
How would
mutations
you know?
they have these 5. Well, I think they do know, right? Because they would have loved to have found, this goes back to the history, they would have loved to find a naturally occurring, Alzheimer's mouse, But yes, they had to make these. They had to make these. You can buy them up in Jackson. I was actually up at Jackson Labs a few months ago.
Oh, right.
Up in Maine.
Yep.
I was very excited. No one else in the, no one else.
The thing is that in these mice, the amyloid forms plaques, right? So, but it's not clear that the catalyst for that is the same as in people, right? So I just don't know if what you find in this model is going to mean anything to people. I was just with that caveat, because it's a really artificial model.
The other caveat, not all T. gondii strains have the same impact in the different mouse models. So there's going to be a lot of challenges here, right? But we're not ready to send people out to eat raw meat or raw lamb or, go visit France, just cure your Alzheimer's. But
Daniel, if we could identify what it is in T. gondii that has this effect that we'll talk about, maybe if we could identify a single protein of T. gondii, maybe that would be therapeutic. But not, I have a problem with infecting people with T. gondii.
I think we talked about that in the previous T. gondii Toxoplasma paper, didn't we?
We don't seem to have much of a problem infecting people in age.
Your colleague
Just go to a restaurant.
In Glasgow, she wants to express proteins in the brain using T. gondii. It's the same concern, right? You're going to get that.
I don't think she wants to, I think that was just a paper, a proof of concept paper. So I don't think it's in the pipeline, I would say.
Well, she said we're just trying to see if it works, and then we'll worry about the other things later. But
exactly.
Daniel, just on the topic of the mice, do we not have any animal models at all that develop amyloid deposits in the brain at all? Did you know? Because I couldn't really find any information. I sometimes hear that dogs can get Alzheimer's, but I wasn't sure.
Yeah, no, this is really the model. I did a little work myself on the idea of isolating antibodies, right? So it was single cell cloning of tau. I was in the tau camp, you could say.
Camp.
And there is some interesting, and again, they use the same mouse model where you have antibodies that target the tau protein. You treat the mice with these antibodies, you reduce the tau protein in the brains of these mice. But yeah, this mouse model is really the workhorse, but that's a problem. It's a mouse. It's not a person. Yeah, I mean, it's good and bad, right? It's not a primate. I'm always a little concerned when they start using primates and giving them, dementia, cognitive impairment. So, yeah, unfortunately, this mouse model, for all its limitations, is really the workhorse.
That's right. It's worth mentioning at this moment that when mice become infected with Toxoplasma in nature, it's like a self-fulfilling prophecy. They lose their fear of cats. And this has been a conundrum for people to understand why that happens until they finally found out that Toxoplasma interferes with olfaction, probably when it affects the region of the brain that receives the messages from the nose. And the thing that they're most unaware of is cat urine.
Mm-hmm.
And that is the tip-off to a mouse to run the other way. And mice with toxo do not perceive that, and therefore they become eaten, and therefore they transmit it to the cat. You've got these teleological arguments that don't make much sense unless you just say by random mutation this came about. And it is now a self-fulfilling prophecy to infect cats with unafraid mice. So people jumped on this bandwagon for a while too and started to, if it can affect a mouse's ability to smell cat urine, maybe it affects the way that people sense each other. And maybe our olfaction is affected as well, but in ways that we're not totally aware of, unless you're a gourmand or a wine taster of some sort, where your sensations are acutely experienced. But I don't think any of that data has any grounds at all for continuing this discussion any further, except that mice become an exception to the Toxoplasma rule when it comes to smelling cat urine, rather.
All right, so let's jump into, so what do they do? So we got all this background, everyone's excited. So now they're going to take these mice, the 5X fad, right? So that's our particular Alzheimer's mouse with the 5 mutations in the familial Alzheimer's disease gene. And they're going to, and this is interesting, they're going to intraperitoneally infect 12-week-old mice in the first set of experiments. So that's the first kind of interesting issue that I had is I understand the ease and sort of going ahead and intraperitoneal, but we're now stepping even farther away. Like, well, why not oral? Why not, why not the way they acquire it usually in nature? Why not the way that human beings would acquire it, right? So
maybe it's more quantitative if you inject them intraperitoneally, whereas with the oral, you would have to feed them cysts, wouldn't you? Or no, that they actually get infected with oocysts, don't they, the mice? Yeah.
So they use tachyzoites here. Yeah. What would you be ingesting normally?
The Bradyzoids.
The oocysts that would come out.
The mice would ingest oocysts.
Oh, they could. That's true. They could.
Yeah. So let's go to the figures, and these are great figures. I don't know if
so they wait 6 weeks, right? But they say, I don't know why 6 weeks.
That's what they're going to do. So they're going to. We'll go to figure 1 for those that are following. I think this is open access, right?
It is not now.
Oh, that's a shame. So we've got to figure out how to. We got to get rid of this. Like, people can't access science, right? I mean, this is exciting stuff, and I'd love people to read it. But so they start off with figure 1, and it's really pretty straightforward. What they're going to do is, as mentioned, they're going to take these mice, and they're either going to give them PBS, they're going to give them saline as placebo, right? Apparently that's acceptable as a placebo. Or they're going to infect them with the T. gondii. And then at 6 weeks post-infection, they're going to look at the braids, and they're going to really quantify the amyloid density, plaques per area, basically. And they have some beautiful bright colored pictures, but then they also have some nice, I actually kind of like the graphs. You see the individual mice on the graphs, or the individual amyloid density results. And you can see that there's a significant difference. I'm going to say a significant reduction in the amyloid density per brain region in the T. gondii infected mice.
Yeah, no argument there. Yeah.
Yeah, very, very clear.
Even RFK Jr. would agree with you.
They wouldn't let you do this. It's infectious disease research.
Yeah, you got to stop this. But maybe maybe you could say I'm studying Alzheimer's. I just happen to be looking at T. gondii. Maybe I can get away with curing people from getting dementia, right? But then the other, and this is I guess what Dixon sort of started off. Why are we talking about global and regional issues? And this gets into figure 2, where they're actually going to look at all these different, and I'm going to say all these different. I mean, what is this? Dozens of different regions of the brain, and they're going to quantify the amyloid density in the PBS, so the saline placebo treated versus the T. gondii infected. And you can say the vast majority of parts of the brain, and sort of pick a few out at random. The olfactory, right? We see a clear difference in the amyloid density in the olfactory region. A clear difference in the gustatory regions, right? The part that affect taste. Significant impacts in somatosensory, in cerebral cortex. Not so much when you get down to brainstem, and thalamus looks pretty similar there. Hypothalamus, cerebellum, things like that are pretty. So not only are we seeing overall significant reduction, but we're also seeing significant reduction in particular areas that have behavioral significance as well.
That's it.
That's it. That's the data. So then we get into our discussion. Really straightforward.
One picture is worth 1,000 words.
So Daniel, in the discussion, they do say no study have shown a protective effect of T. gondii infection against dementia in humans. They've looked.
Yeah. Notably, there are no studies. And what I say, it should also be noted, thus far, no study has investigated the Alzheimer's disease pathology during life or at end of life in humans seropositive for T. gondii infection. Right. So there's really a lot of work to still be done.
But the problem is that, here in this mouse, they infect for a defined period of time and then look for amyloid, right? And when would you do it in humans? Because it probably starts to develop. When does typically Alzheimer's manifest symptomatically, Daniel?
It's really going to be 70s, 80s, right? So it's going to be, as they use this sort of cutoff as over 60 when they're talking. And there's this idea, right, that, at least a suggestion that this is not necessarily, okay, let's say you're like Dixon described, you're in France, you're eating your lamb tartare as a, 1.5, 2-year-old. And now you've got dormant bradyzoites just sitting there. What is it, 70, 80 years later? Are they're really going to do the work? Or do you need a fresh infection, which is going to trigger the migration or activation in the brain of certain phagocytes, certain macrophages that are amyloid plaque averse, I guess I'll say.
Yeah.
Because that's the model. The model idea is you infect the mice. It triggers these macrophages. The macrophages are consuming and clearing the toxic amyloid protein. So it becomes this other issue. So let's say you continue down this road. You've got an individual, they've started to develop Alzheimer's. People are pretty desperate. I mean, people actually commit suicide when they realize that they're headed down this road. So
that's right.
If someone is, we'll say, toxoserology negative, and they rare lamb, which people do all the time. Like, is this really like mad science stuff to say, give it a try and see what happens? Or if they're toxoserology positive, but it's a really distant infection, are they now not going to have any?
Yeah, the timing is the key probably, right?
Right.
And if you might even want to, and if you're going to infect people, you might want to do it before symptomatic symptoms are evident, right?
So that's the interesting. In the mouse model, they take mice once they start to get symptomatic, they infect them, and they actually for 10 months in a mouse, which is 20 years in a person, I'm going to multiply. They get better. They have improved memory and learning and cognition. So they've already waited till they start to have early symptoms, and then they're reversing the process.
So what I could see, Daniel, is that you have someone with advanced Alzheimer's. It's like advanced cancer. There's nothing that can be done. And you can do a clinical trial and feed them toxo and see if they get a reversal of symptoms.
Yeah. Yeah. Some of them get, real steak tartare made from the cows, and some get the steak tartare from the lamb that we know.
Wow, that's a great control. That's a great placebo.
That's right. Well, you have to make sure that they have an intact immune system, however, because I think if they're immunocompromised for whatever reason, and the older you get, the less immune competent you are.
Yeah.
Then you could never do those studies again after you do them in those people, because a lot of them will die from the infection.
Yeah.
Oh, there's just so many ethical issues with these experiments.
Yeah. No, you are right. You are absolutely right.
The thing is, though, with such a lethal disease, people are always desperate to try things, right?
I mean, it's devastating, right? I mean, I would say personally, right? If I was at that point, I'm in my, let's say I'm 78 years old and I start to develop, confusion, dementia, I get diagnosed with Alzheimer's.
And you just became elected as the president of the United States.
I don't know what worm I had in my brain prior to this, but But yeah, no, I mean, it's, and I think the challenge here, and we're going to talk about a paper in the future about trichinella and the microbiome, where it may not just be some simple protein. I mean, here it may be that the toxo-infected macrophages are somehow triggered, and it's much more complex. It isn't just finding one thing you inject that will then activate those.
Do you think you even need infectious toxoplasma?
So I don't know. I mean, that's a curious thing is like, what does it take in, and maybe that's where you work a little harder in this mouse model.
Yeah. I mean, because if you show that you can inactivate it and do the same thing, then it's a much easier trial to do in humans, right?
Yeah, so there are irradiate them.
Yeah. There's a wonderful set of experiments that was done by Elmer Pfefferkorn at Dartmouth to develop a vaccine in cats. And what he did was he isolated temperature-sensitive mutants, which would not grow at 37, but would grow at room temperature. And you can do that very easily because they'll infect any cell, remember. So they'll infect B cells. And any cells that, in other words, he got his strain of toxo. But, if you were to inject that into a patient, and they found their way into the deep tissues and then died, that wouldn't be a long enough time to see whether or not this infection had an effect on the outcome of Alzheimer's disease. You'd have to do something like an inducible gene for temperature sensitivity that you could raise the body temperature of a patient anytime you wanted and kill off the infection in case it got out of control. And that might be ideal. That may be the way to do this experiment, because when people do volunteers for malaria, they give them not a drug-resistant strain, obviously. They were all working on drug-sensitive strains because if it gets out of control, of course, then they will give them the drug and cure them of the infection. In this case, though, the bradyzoites don't do much. They just sit there. They must do something because they're affecting the outcome of the amyloid. Maybe indirectly, though. And so a few proteins, they may be metabolizing a few proteins or a few environmental cues that triggers the invasion of macrophages. And once the macrophages are there and they're looking around, they don't see anything because there's no antigenic signature of this parasite because it's not dividing. Then they say, oh, the amyloid, what the hell? We might as well eat as long as we're here. So they have a picnic and they devour all the amyloid. I'm simplifying it, but they don't end up killing the toxo. That's the one thing they don't do because of this remarkable survival characteristic that the organism has evolved in order to live long and prosper, basically. He wants you to live long and prosper and then die. Just don't get cremated, that's all. Otherwise, there goes the infection.
But I think that's it. I think pretty straightforward paper.
It's
still a lot
fantastic.
More work to do.
It's fantastic.
Okay, then let's move on to our hero from Christina.
Let's do that. But first I want to say thank you, Daniel, because actually the paper, I needed to read that with my dictionary of brain anatomy. So it was really well presented. So I think that will be really appreciated by our listener. So I want to talk today about a veterinary parasitologist. So we mostly really talk about human parasitic infections on our podcast, but of course animals are also parasitized, and parasites can make animals as sick as they can make us sick. And then if we consider the livestock industry, for example, veterinary parasites can also have a really tremendous impact on food production. So I think it would be an important topic to maybe consider in the future. So the veterinary parasitologist I'd like to cover is a German parasitologist with the name of TWiP. So for those of you who are able to read German, there's a short Wikipedia entry for him in German. But for everybody else, I have copied the link for an obituary in English into our show notes, which was published in 2023, shortly after his death. So TWiP, he was born in 1929. He grew up in the Thüringen region in Germany, I think central Germany, if my geography memory serves me right. I have no information about his childhood other than he grew up on a farm, and I presume that this is where he came across many parasites, provided that his farm was actually a livestock production farm and not, wheat or oats or something. Anyway, so I presume he was exposed to the effects of parasitology as a child on the farm. So he went on to study veterinary medicine in Leipzig in 1948. That took 5 years, and he remained in Leipzig. He was first employed at a local animal hospital, and then he was hired as a chief veterinarian at the Leipzig Zoological Garden. And it's really then that his interest in parasitology became apparent. So he started working on the control of ectoparasites, but also helminth and coccidia of farm and zoo animals. His primary interest really was small ruminants, in particular sheep. So I just want to digress briefly, really, to talk about the coccidia in the farm and zoo animals. So this is maybe a parasite that we are less familiar with. I just want to say just a couple of words about it. So coccidiosis in ruminants, and actually also other animals, chickens in particular, and cows, is caused by parasites of the species Eimeria, which is an intracellular protozoan parasite with a quite interesting life cycle. well, all parasites have interesting life cycles, really, if we're honest. And these parasites can cause really considerable disease and economic loss. So the kind of primary clinical symptoms in an animal that is infected with Eimeria is probably diarrhea. So the severity can vary from self-limiting to severe. And in this case, the animals can die very quickly. If the animal does survive, the survival can be associated with reduced fitness of the animal because of the permanently damaged gut tissues. The Eimeria, they live intracellularly inside intestinal gut cells, and they kind of replicate, not dissimilar of malaria, really, their life cycle. I don't really want to go into too much detail in the life cycle because that would take too long. But so essentially when so the infection is acquired really orally, and these are oocysts that are taken up. So just like in the Toxoplasma that we just talked about, and sporozoites are released inside the mammalian host, and they invade intestinal cells, and then they go replication cycles, so they develop into schizons, and then they generate merozoites, and you're probably familiar with those names from the malaria parasites. And these merozoites, they can infect more cells, and eventually gametes develop, and then a sexual phase happens, resulting in new oocysts, which rupture the host cells, and then the oocysts are released in the human. So this is really just the short form of the life cycle, and that's really what causes the clinical disease, these ruptured intestinal cells. And the symptoms really vary a little bit by where the infection occurs. So some Eimeria prefer infecting small intestinal gut cells, others prefer the colon to infect. So the clinical symptoms are a bit different depending on that. Anyway, I suppose I should go back to our hero before I get carried away too much. So Theodor Hippe, he actually became very young. When he was only 31, he became the chair of parasitology and veterinary zoology at the Faculty of Veterinary Medicine at the Humboldt University in Berlin. And he initiated a really broad research agenda and established 4 working groups, one focusing on protozoa, then one on helminth, and then another one on entomology, and finally diagnostics. He was working there for 35 years, so he was really steady, and he remained there and he focused. Particularly, he was really interested in translating laboratory research into parasite control that could be taken into the field. And he was also interested in immune processes of parasite infections. So under his leadership, diagnostics of parasitic diseases was a really big mandate in the institute. So, and really, diagnosis is really a first step into kind of finding out where parasitic infections occur, and that can then help to develop control and prevention strategies, I suppose. So at the institute, Theodor Hippe, he developed integrated parasite control approaches which combined hygiene measures, presumptive treatment with antiparasitic drugs, and on-farm training. So, how farmers can maybe avoid infection, or, maybe different grazing strategies that could be employed, moving the sheep around, for example, so that they don't become exposed to oocysts that have been shed. So that's really what he wanted to do. Actually, one important piece of research he led resulted in the control of the warble fly, which had a really, really large impact on the livestock industry. And really, he kind of did really big surveillance studies on hundreds of thousands of heifers and cows to understand the infection dynamics of the warble fly and also treatment efficiency, and developed a mathematical model to really what was the foundation of a control program that led to elimination of warblefly in Germany. He also had 2 parasites named after him, Eimeria hippii, which is a coccidian parasite of the American mink, and the Magutia hippii, which is a filarial worm in a gecko in southern Africa, which is transmitted by sandflies. So, he got his name on parasites. He wrote many textbooks, particularly on sheep, and also a really seminal textbook on parasitology, veterinary parasitology. But he was described, and I'm quoting here, an immensely intelligent man and an outstanding scholar, and his passion for parasitology and his insights into parasite host dynamics inspired generations of early career scientists. And he died quite old, at the age of 93 in 2022. So I thought this would be a slightly different hero of parasitology from what we usually talk about. And please go and Google the warblefly, because it's really interesting. And also Google Eimeria, also a very interesting parasite.
Thank you, Christina. Thank you very much. It's at TWiP 248. You can find show notes at microbe.tv/TWiP. You can send us questions or comments, twip@microbe.tv. And if you like our work on these programs, please support us, microbe.tv/contribute. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, thank you. And everyone, be safe.
Dixon Despommier, trichinella.org, thelivingriver.org. Thank you, Dixon.
Thank you, Vincent, and everybody else, and have a happy Thanksgiving to those that celebrated.
Christina Nowla is at Glasgow University. Thank you, Christina.
Thank you for having me. I'm not celebrating Thanksgiving, but I wish I did. Because
no, you're perfectly welcome to.
I suppose I could.
Yeah, go to Dixon's house. He'll be happy to have you.
That's absolutely right. We got plenty of room here.
I'll fly right over.
Oh, good.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
Another TWiP is parasitic.
That was seamless, Dixon. Very good.